Hi, my name is Vincenzo Bonnici. I'm an assistant professor with the track at the University of Parma, and I'm also a member of the Infomics Laboratory of the University of Verona in Italy. So today I would like to talk about capturing sequence homology in microbial pangenomic analysis, and more in particular about the computational tools that is called PanDelos. So PanDelos has been recently presented at the BBC conference in 2011, and it was then published in BMC Bioinformatics the year after. This is the paper regarding the work. So what we are talking about? We are talking about pangenome. This term was firstly introduced by Tetlin and colleagues in 2005 for describing the complete set of genes, namely gene families, that are present in a given species, in this case a bacterial species. In fact, they examined the 7 strains of Streptococcus lactei and divided the genes that we discovered the gene families into 3 groups. Core genes, that are genes that are shared, so they are present in every individual of the study, so they may represent core functionalities of this type of organs. Accessory genes that are shared by at least 2 of the study isolates, so they may represent non-essential functionalities that is shared within a group of the organisms, and singleton genes that are genes present in only on the studied genomes. So they may represent specific functionalities owned by only one of the strain of the isolate. Bacterial studies, the understanding of the effective impact of the gene within a population arises in clinical applications. For example, to identify that drug targets in vaccines and in the bacterial study. So in this perspective, this type of analysis, genomes are represented as set of genetic sequences that are just sequences of the genes. And the goal is to detect the homology among genes that belong to different genomes. Functional homologies usually relates to the conservation of a biological function, but here we are interested in sequence homologies, that is the conservation of the genetic sequences. In this context, there are 3 types of homology: orthology, that are genes that are vertically transmitted from a parent to a descendant; paralogy, that is the one of more copy of the same gene within the same genome; and xenology, that is a gene that has been mainly produced via horizontal gene transfer. Horizontal gene transfer is a widely diffused behavior to exchange genetic materials among microbes, but among every type of organisms. It happens between closely related genomes. It also happens within very distant related genomes. So this was changed our perspective of how the organisms are genetically related to each other, such that what we can think about the tree of life is no more a tree, but is a kind of complex networks in this genetic material transfer between all the living species. So how it is made common pipeline, computational pipeline for pangenomic studies in microbial organisms. Usually we start from a set of genomes that are in the form of assembly of contigs. So for each genome we know the complete sequence of the genome. And it is an annotated sequence. It means we don't know which are the genetic sequences within this genome. So we have to run a gene detection algorithm to detect the location of the genes within each genome. So in this way, in this example, we have 6 genomes from A to F, and after the gene detection, we have detected all the genes that are present in this example. We know that if a gene has been transmitted to another organism, more probably the sequence of this gene will not be 100% the sequence of the original genes, but there will be some variations in these genes. So, for example, the yellow gene that is present in every of the genome of the analysis is in a kind of variated form within all of the genes, within all of these genomes. So the goal of the pangenomic pipeline is to run a clustering by sequence homology in order to understand which are the gene families that are present in every of the analyzed genomes. So they can be called core. The gene families, for example, the green one that are present in just a few of the analyzed genomes, in this case 4 over 6, so they can be called accessory, and the singleton genes that are genetic sequences that are present in only one of the analyzed genomes. All of these information represent the pangenome of the group of genomes that you are taking into account. Starting from this information, we can run downstream analysis, or we can study biological functions. We can extract biological meaning from this information. For example, what we can analyze is the pangenomic distribution of the gene families. So on the x-axis, we put the number of genomes, and we count how many genes, namely gene families, we have found in that specific, to be present in that specific number of genomes. So for example, here we found 4 genes that were present only in one genome, so that were singleton. On the contrary, we found three gene families that are present in every of the 6 of the analyzed genomes, so they can be called core genomes for our analysis. And in the middle, we have all the accessory genomes. Usually, we can say that this type of U-shape for this distribution arises for pangenomic analysis. So we have a relatively high number of singleton genes, but we also have a relatively high number of core genes. What we can also study by means of this information is the closure of a pangenome. So we can say that a pangenome is closed when we know enough information about these pangenomes. That means that if we add another genome to our analysis, the number of new genes new gene families that we discovered by adding this new genome is relatively low. In fact, here we can see that usually on increasing the number of genomes that we put in our analysis, the number of new genes decreases dramatically. So in this case, on the left, we have a study in which we can consider the pangenome closed, because there is the line which shows us the number of found gene families. There is a plateau on this line, and we can decide whether it closed. On the contrary, on the right image, we can say that this type of pangenomes that we are taking into account is not closed, because we still not have a plateau on the number of genes that we have found. So we still need to sequence more genomes for that strain, for that species, or for that group of genomes in order to understand enough about the pangenome of that group. So PanDelos is a computational pipeline. It is an alignment-free methodology for discovering pangenomic content among complete genomes. Complete genomes means that we know the complete sequence of the organisms. So it cannot come in the forms of fragments that still have to be assembled. In this way, we are able to detect every single gene that is present in the genome. Sequence similarity is then calculated by comparing k-mers of the genetic sequence. So given a sequence, a k-mer is a subsequence of length k, and usually the problem is to understand which is the right value for k in order for our analysis, in order to get good results. Then a bidirectional best strategy is combined with a community detection algorithm in order to obtain the final set of gene families. So I will give more detail about this in the next slides. PanDelos is mainly designed for working with genomes that are very distantly related to each other. So it can also be applied to genomes that are close, evolutionary close to each other, but it works well with this type of genomes. So this is the main a figure that represents the entire pipeline, starting from annotated genomes. We can use which tool we want to annotate the genome. So once we have the set of genes, we can start with the pipeline. Initially, there is the choice of the optimal value k, namely the word length. And then there is a selection to filter gene pairs comparison. Then we calculate for each pair of genomes, we calculate the similarity between each pair of genes of the two genomes. And on top of this similarity, we can detect the bidirectional best hit. From the bidirectional best hit, then we go ahead. So the choice of the right resolution is performed by taking into account a theoretical law that comes from an application of information theory to genomes. In this paper, we discovered that the right resolution to analyze the genome is k equal to the log base 4 of the length of the genome. And this is the right resolution to show the difference between a real genomic sequence and a random sequence of the same length. Here we adapted this idea to a pangenomic study. So instead of having one genome, we have a set of genomes. So as length of the genomes, we take the complete length of the pangenomes, so the sum of the length of the genes, and we calculate the logarithmic base 4 of this length, and we take this value as optimal word length, so as an optimal resolution for our analysis. Then we perform all versus all sequence comparison, but before we run a selection by dictionary intersection coverage, which means given two genes S and T, so we compute their dictionary, that is the set of k-mers that are present in each of these two genes. So if we compute the intersection between these two sets, this intersection must have a minimum amount of 2 over k, where k is the word length distribution. Then we also apply a similarity threshold, that is apply another filtering for the similarity threshold, that is simply we avoid to compare genetic sequences that is 1/2 of the length of the other, just to avoid unuseful computation. The sequence homology, so the sequence similarity, is computed by means of a generalized Jaccard similarities, that in this case, given two sequences, for each k-mer that appears in one of the two sequences, we sum the multiplicity, so the number of occurrences of that k-mer of within that sequences, the minimum of these multiplicities as the numerator, and we take the maximum of this multiplicity, we sum it at the denominator. Which means also that if a k-mer is not present in one of the two genomes, the minimum multiplicity is 0, so it sums on the numerator, and the multiplicity on the sequence where the k-mer is present, it's summed up to the denominator. So this is a metric, and it is in the interval 0, 1. So it is normalized, which means that it is a very good measure to be used. So usually it takes similar sequences in the scores between 0.1 and 1, and it assigns scores similarities close to 0 for the similar sequence. To compute this similarity, we adapted an efficient framework called EG-tools that was reused in computational analysis of DNA sequences. And here we adapted it to analyze the genes as DNA sequences, but also to analyze them as sequences of amino acids. The framework extends the enhanced suffix arrays with an additional array that is used to solve the document listing problem. So the document listing problem says that if we have a series of documents and we want to know, given a word, in which document this word is present in an efficient way. Okay, so we adapted this kind of data structure for our for our purpose, and we can show that every step of our analysis regarding sequence homology similarity can be performed in linear time thanks to this kind of data structure. Then for each pair of genomes, we have to compute bidirectional best hit. So a bidirectional best hit is a pair of genes such that one genes belong to one genome, in this case the blue genomes, and the other genes belong to another genome, in this case the red genomes. So they are in a bidirectional best hit if they are each other, if they are reciprocally the most similar sequence from one genome to another. So in this case, we take these green genes, we can say that there is a very low similarity with these three genes, but it has a similarity of 1 with these other 2 genes of the second genomes. So since it's reciprocally the same also for these green genomes, this specific pair is considered the bidirectional best hit. On top of the bidirectional best hit relationship, we build an homology graph. Such graph is built such that the vertices are the genetic sequences, namely the genes, and the edges are the homology relationships, namely the bidirectional best hit. Given the graph, the homology graph, we can discover connected component within this graph. So a connected component is defined inconsistent if it contains 2 edges belong to the same genus that are not accounted as paralogs. In fact, before this step, we have to take into account that we do the same 2x2 genome analysis, and in this, so for example, here we compare the blue genome with the blue genome itself in order to discover the paralogs, and there is an ad hoc procedure for discovering these paralogs. So in case of inconsistent components, the effect is that we have some additional edges that we want to remove, because an inconsistent component usually is aggregating 2 gene families. So we need a way to break these connected components into 2 different connected components that represent, each connected component represent a given gene families. To do so, we use the Gierman-Newman algorithm, that is an algorithm for community detection. It calculates the betweenness centrality among the components of the network, and then progressively removes the edges with the highest betweenness centrality. So in this case, we can see in the example that the 2 red edges will be deleted because they will have the highest betweenness centrality. In fact, they connect these 2 very dense part of the components, and they are a bridge, kind of bridge between these 2 parts. So at the end, the 2 red edges will be discarded. And as a final step of the analysis, after discarding the edge, we obtain the resultant gene families. In this case, we have orthologous and paralogous genes within the same gene families. Each component is a gene family. We have dispensable gene families and singlet of gene families. So at that time, there were a few computational methodologies for pangenomic analysis. During the time, many of them have been published. This is a table that was made in 2021 regarding the computational analysis. But let me say that it's very difficult to perform the statistics, to understand the performance of these tools. At the time, Rory was the best, the most used software for this kind of analysis. It combines alignment-free approach. In fact, we use a clustering approach by means of K-mesh multiplicity, by means of CD hit, with an alignment-free measure that comes from BLAST sequence homology, that is then combined and used into clustering community detection algorithm called MCL, and it combines these 2 pipelines to obtain the final set of gene families. Edgar, the problem with Rory is that CD hit and blast, for example, CD hit, it requires the user to specify, CD hit use a fixed k-mer length that is usually 5 or 6, but the user can specify it. And for blast analysis, the user have to specify some threshold, for example, the sequence identity and E value in order to consider two sequences as the same. And this is as user-chosen parameters, and when you have user-chosen parameters, it's always a risk because you don't know if these parameters are right for your analysis or not. So the author of Edgar developed an adaptive score threshold. In fact, it firstly computes the distribution of the blast-based score, normalized by the sequence score itself, and then it's trying to understand which is the best cutoff to be used in order to consider two sequences as similar or not. So for our test, we used a use case based on the mycoplasma. Mycoplasma is a genus of bacteria that lack as a cell membrane lack a cell wall in the cell membrane. It is highly heterogeneous, an highly heterogeneous group of bacteria that is involved in many diseases, and for this case, it is also considered the reference case in pangenomic analysis. It is also a reference genome for building synthetic minimal genomes because it is the smallest bacterial cell yet discovered. So we analyzed the coding sequences of 64 individuals belonging to 27 different species. And this set composed the list of mycoplasma isolates that were at that time present in the Edgar website. So since Edgar is not a standalone pipeline, we have to use the website, and we can only run on the isolates that are already present in the website. These were the 64 individuals that were already present in Edgar, and we used them. So these are some statistics regarding these individuals: the genome length, the distribution of the genome length. The average is 894 kilobases. The number of sequences for each genome, so how many coding sequences we have for each genome, and on average we have 740 coding sequences for each genome. And the gene length distribution, so the length of the genes that we found in this group. So the average length is 214, but we can see that there are some genes that are also, which length is over 4,000. So, but in general, we are analyzing this kind of groups. So the total number of coding sequences were 47,385. So this is a heat map which shows the genomic distances within the 64 analyzed organisms, and the genomic distance or similarity was obtained by means of the CB3 software, that is a well-known software for computing genomic distance, and is based on the concept of k-mer. By running Rory with default parameters, which means a threshold of 95% blast identity, we found no core genes for this analysis. Then we modified this threshold. We use a 60% of blast identity, and and Rory at that point was able to find the first core sequence of our analysis. So in total, we were able to find 2,000 more different gene sequences. One was core sequence, and many of them were singleton sequences. Regarding Edgar, instead, we found 9 core genes by means of Edgar's, and a total of 15,000 distinct families in the analysis. The core genes, the annotation for the core genes were this one. Many of them were ribosomal proteins. By running PanDelos, we found an optimal word length k equal to 5. So the total number of 5-mer was then more than 1 million. And interestingly, we found that there is the number of k-mers that appear in at least 2 sequences is relatively high, which means that there is always between 2 genetic sequences there always be a k-mer that is in common, at least one k-mer that is in common between these 2 sequences. A detail that here we do not analyze the DNA sequence of the genes, but we take into account the amino acidic sequence of the genes. So overall, we found 5,000 singleton genes for a total of 40,001 analyzed sequences. So in this was the before the homology network after the bidirectional best hit, and then we refined this homology network. So overall, we found 6,720 clusters, namely gene families on the homology network. So they are what we can define the gene families. Of these, 258 were singleton genes, and 6 gene families were found in every of the analyzed genes, so they were considered core genes. So if we compare with the result of Rory, that was just 1 core gene, and with the result of Edgar, that were 9 core genes, we can say that PanDelos was able to discover more core genes. We also ran a functional analysis of these 6 core genes in order to understand if we did not put together 2 core genes, 2 core families, or if we did not split the core genes. And the functional analysis say that these 16 core genes, the genes core families, the genes within each family, they share a known functional biological function. So we also ran the 3 software on other organisms, rather than mycoplasma, that were the type I, that is the serotypes of the Salmonella enterica. These are some statistics regarding the type I, Escherichia coli, for some of the species, it is a species of Escherichia, Xanthomonas. So it is a genus. We ran at the genus level. So we tested our software also at the genus level. And as a result, we obtain this kind of curves for each of these analyses. And as we can say, PanDelos is almost always the software that is able to find the highest number of core genes. What is the problem here? The problem is that we don't know if this curve, so the result of this analysis, is true or false, because we don't know which is the reality of the gene family composition in this genome. So for this motivation, in the next talk, I will talk about how to systematically benchmark this kind of source. So for today, I thank you for the attention, and see you on the next video.